Hello, my name is Tatiana Farud. I'm the principal investigator of the National Cell Repository for Alzheimer's Disease, also called NCRAD, and we're hosting a webinar today describing our services for incoming samples. NCRAD is located at Indiana University at the School of Medicine, and we are funded by the National Institutes on Aging, and the website is www.ncrad.org. Our outline for today is we'll begin by discussing biospecimen uses and research. Then we'll discuss NCRAD services. So let's begin with uses for biospecimens. As you can see here, we have the most common specimens that we bank here at NCRAD along the left, and then we discuss the source of the sample and also its uses. So let's begin with DNA. DNA can be obtained from blood, from other kinds of tissue, and saliva. Frequently, we'll obtain DNA from brain tissue at NCRAD as well. DNA can be used for genotyping of particular variants, including ApoE. It can be used to study genome-wide SNP arrays, or GWAS. It can be used for whole exome or whole genome sequencing, and it's frequently used for studies for epigenetics. RNA can be obtained from blood or from tissue, often brain tissue in the case of Alzheimer's disease. We can obtain from those sources total RNA or microRNA. It's frequently used for sequencing in terms of transcriptomics and can be also used for expression arrays, often called microarrays. Plasma is obtained from blood, and it's commonly used for biomarkers in Alzheimer's disease, such as tau and neurofilament light, also called NFL. There have been inconsistent results with A-beta. Serum is another biomarker source that can be obtained from blood. It's not as frequently used in Alzheimer's disease, but there's interest in developing biomarkers that can be tested in serum. Here we have two additional sample types that we bank in NCRAD. These include peripheral blood mononuclear cells, often called PBMCs. Those are obtained from blood. They're the preserved white blood cells, and they're often used in stem cell research, including the development of induced pluripotent stem cells, often called iPSCs. They can also be used for gene expression profiling. And lastly, cerebrospinal fluid, CSF, obviously not obtained from blood, and is one of the most common sources for biomarker studies in AD, and people study tau, p-tau, A-beta 42, and of course the A-beta 42, A-beta 40 ratio. One of the considerations when considering whether or not to obtain and what biospecimens to collect as part of a study is what would need to be done at the site if that specimen were collected. So across the top, we have blood-based specimens that we discussed on the previous slides, and along the left, various processes that might need to be used at the site. So whether or not the sample needs to be processed on site, as you can see, plasma and serum would need to be centrifuged, but DNA, RNA, and PBMCs would not. Similarly, plasma, serum, and RNA would all need to be frozen on site, whereas DNA and PBMCs would not. They would need to be shipped in bulk, would be plasma and serum and RNA would be possible. PBMCs and DNA need to be shipped on the same day, typically, that they're collected. And the blood volume, if you were to collect all of these samples, would be a little over 50 mils. And you can see in the very last row the type of tube that would be used to collect the different kinds of specimens. There can be challenges in biomarker studies, which makes using a repository like NCRAD valuable. Many factors can affect the performance of a biomarker, and so it's important to collect samples in a uniform way and consistent with best practices. So NCRAD seeks to ensure uniform sample collection, processing, and storage. In order to meet those goals, we have a series of services that we provide for studies banking with us. We provide comprehensive study startup, and this includes the development of a procedures manual. We develop training videos for the study. We perform all site and staff training. We provide sample kits as well as a kit request module through which the sample kits can be requested. We perform data reconciliation. We develop online catalogs, and we will also provide a letter or a budget in support of a grant that's being written where samples will be banked at NCRAD. So let's start with looking at the services that we provide for ongoing studies. So at our website, www.ncrad.org, if you look under Tools for Active Studies, you can see a variety of studies that we are currently supporting. You can also see details about our holiday closures, our shipping address, and handling for Friday blood draws. You can see under that tab the manuals that we've developed for various studies. So NCRAD develops a study-specific manual for the procedures for each study that's banking at NCRAD. The manual contains descriptive and schematic details about sample collection, processing, and shipping. And I'll show you in the subsequent slides some examples of the types of schematics that we develop. Here's an example of the schematic for plasma and buffy coat processing. So in this case, from a single 10 ml tube, we're able to obtain both plasma and a buffy coat, and the buffy coat is used for DNA extraction. So for a site that's going to be using us for the plasma buffy coat processing, we would go over the schematic and make sure the sites have all the equipment that they would need and understand how the sample would be collected and processed. Similarly, for serum processing, again in a 10 ml red top tube, you can see the processing that would need to be done at the site. And here's an example for CSF, where again it summarizes what would need to be done with that sample, how it would be aliquoted, and also, as you can see in step 1, a sample that would also be sent locally for other testing. We have a new initiative here at NCRAD, which will be to expand and provide support for iPSC and fibroblasts. We'll be starting this in the summer, and NCRAD will be developing this service to expand lines that were developed from other researchers. And the goal here is to make it simpler for researchers who've developed these lines not to also have to share them with other individuals. They'll simply send them to NCRAD, and we will expand them for them. A webinar will be scheduled this summer to launch this new initiative. We also provide training videos. These videos describe how samples should be collected, processed, and shipped correctly for each protocol. And those videos are available on the NCRAD website, and we use them for retraining and also for training of new staff. And you can see some examples of those videos on our website. We provide site training. Before sample collection begins, we provide a WebEx training for studies and their study staff. In some cases, if it's a very large study, we'll go to the startup meeting, and a copy of the slide set and a recorded version of the site training is posted on the NCRAD website and is available upon request. In order to build all these kits, we have facilities here at NCRAD where we have all the supplies for our kits, and we're able to put together kits that meet the specifications of a particular study. And this is an example of our kit request module. So for a particular site, the coordinator would come here and they would be able to log in and request kits. They can request kits for multiple visits. They can request additional materials if they run out of something. And as you can see here, all the materials in each kit are also shown on our kit request module. For all of our kits, we also provide pre-printed labels. Some examples are shown here. We provide labels for all collection and aliquot tubes. And as you can see here, each aliquot has a unique number. That's the number in the top of every label. You see where the sample is going to be sent. You see the sample type. And of course, a kit number that links all the samples being collected as part of a particular visit together as coming from that individual and that visit. We also train sites how to package samples for shipment. So we provide extensive training regarding sample packaging and shipment. This does not take the place of site coordinator training that needs to be done on site in order to meet packing and shipping requirements for your own local site. We provide training for the shipping of ambient samples. And we also provide training for shipping frozen samples that would be shipped on dry ice. Here at NCRAD, after we receive the samples, we do sample processing and storage, as you can see on the right. And you can see that we have a number of freezers and also liquid nitrogen tanks where we're able to store samples as well. We perform data reconciliation. What that means is NCRAD coordinators and our IT staff here work together to perform data reconciliation for all the studies banking samples at NCRAD. The format varies by project depending on the data we receive, but we also try to resolve all discrepancies that are identified at login. So we'll contact the site where the sample was collected and immediately try to resolve any issues that we see on forms, on sample labels, or anything that doesn't meet the requirements of the study protocol. Examples of queries that we run would be missing samples, incorrect samples that have been collected or shipped, damaged or incorrectly prepared samples, unlabeled samples, samples that are labeled with incomplete information or mislabeled samples, and we'll also flag discrepant information that's documented between the NCRAD biological sample and shipment form and the data entered in the main study database, typically at the Data Coordinating Center. We develop online catalogs as well. These catalogs are online for researchers who want to easily identify samples that they need for their research. These can be queried by approved researchers to explore the range of biospecimens that we have available at NCRAD. And we have a second webinar that we've recorded that you'll be able to view as well on our website, where you can learn more about requesting samples. If you're preparing a grant in which you'll be banking samples with us, we would ask that you contact us, and our contact information is right there. Do it as early in the grant planning process as possible. We'd be glad to discuss with you the samples you're interested in collecting. We can provide you with a quote that you include with your budget, and we can also provide a letter of support that you can include in your grant. So now at the end of this webinar, if you would like more information or have questions, please contact us at altstudy@iu.edu, or you can call us toll-free at 1-800-526-2839.